So good evening, everyone. It's a real honour and a privilege for me to be here tonight with you to give this Sir John Monash lecture, and I'd like to thank Professors Mahindran and Gan for the invitation to be here and present tonight. So I'm going to be talking tonight about malaria, and more specifically, I'm going to talk about how partnerships are working to try and discover new drugs to treat this disease. So just before I start, I have about a minute's worth of slides that I just wanted to play. I won't speak, I'll let you read them. And this is for the benefit of those of you that may not know an awful lot about malaria, and these are some statistics that were taken from the latest edition of the WHO World Malaria Report. And I think this final slide really illustrates the true tragedy of malaria. This is a preventable disease, and it is a completely curable disease if it is diagnosed properly and treated properly at an early stage. However, there has been considerable progress in the fight against malaria over the past 15 to 20 years, and there have been some new targets set for the elimination of malaria going forward. So you can see on this slide, in the graph on the right, that the number of deaths due to malaria has decreased quite substantially over the period since 2000. And as reported in the 2016 WHO World Malaria Report, over the period of time from 2000 to 2015, there was a 41% reduction in incident rate and about a 62% reduction in mortality rate. And over this period, it was estimated that there were approximately 6.8 fewer malaria casualties because of these trends, and that's relative to the incidence and mortality rates that were in place at 2000. So that's a very substantial saving of many, many lives. Over this same time period, there were about 17 countries that actually eliminated malaria, so another success. So this progress can be due to many, many factors, and most importantly, or one of the most important factors, is that there has been a real increase in the level of political commitment as well as funding that has been put into malaria control programs. That, in combination with the use of improved vector control strategies, most notably the use of insecticide-treated bed nets, as well as indoor residual spraying to reduce the transfer of mosquitoes and malaria via those mosquitoes. We also have much more improved disease management through better diagnostic tests and treatment strategies, and we have better strategies for chemoprevention. And all of these factors combined have really contributed to this significant reduction in mortality that we've seen since 2000. However, the report that was issued late last year by the WHO, the 2018 World Malaria Report, highlighted some fairly alarming trends, and they were that over the period from 2015 to 2017, there is actually an increase in the number of cases of malaria worldwide, and we haven't seen this trend for quite a while. Between 2016 and 2017, there were 20 countries in the world that reported a greater than 20% incidence in the number of cases of malaria. And similarly, the rate of reduction in the mortality has slowed since about 2015. So these trends and these statistics have a number of experts around the world quite worried that we might actually be starting to see an increase in malaria incidence once again. So these statistics are also very important when you think about the ambitious targets that have been set for elimination. And in 2015, the WHO launched what's known as the Global Technical Strategy for Malaria for the period of 2016 to 2030. And this strategy targets 90% reduction in both the incidence and mortality rates relative to the figures that were present in 2015. The strategy also is targeting elimination in more than 35 countries that were actively transmitting malaria at 2015. There's another plan called the Aspiration to Action Plan that was launched by the UN Special Envoy for Malaria and the Bill and Melinda Gates Foundation that takes these goals one step further and actually targets complete malaria eradication by the year 2040. So these are ambitious and many think quite difficult, but yet achievable goals for us to try and strive for. So in the Global Technical Strategy, there were also highlighted some milestones for 2020 and 2025, and you can see those listed here on the slide. And the latest data that's been reported suggests that in terms of the mortality rate and the incident rate, we're really not quite on track where we want to be, and if the current trends continue, we may not actually achieve those goals. However, if we look at the elimination and the countries that are targeting elimination, the target was for 10, and I think this is well on track to be achieved by 2020. So this map just was published in a paper in PLOS Medicine in 2017, and it's a map of the world that shows the transmission status of various countries at that time period. And you can see the countries in green are the 91 countries throughout the world that were actively transmitting human malaria. The countries shown in purple were those that were certified malaria-free since about 2007. And the countries shown in orange are the 21 countries that have the potential to eliminate malaria by 2020. And if you look closely at this map, hopefully you can see it, it's a little bit small, you can see Malaysia is one of those countries that is shown in orange. So according to a report that was published, the 2019 progress report for the WHO E2020 initiative, this is an initiative for the 21 malaria eliminating countries. If you look at the data that was published in this report, it suggested that there was a significant reduction in the number of indigenous human malaria cases in Malaysia, and in 2018, the preliminary data suggested that there were no cases of indigenous human malaria species that were reported. So this is really a fantastic effort, and it's ahead of schedule. You can see in the table on the right, there has been a significant decrease in malaria case incidence over the period from 2000 to 2015. And this is due to a number of different factors, government funding, government commitment, improvements in diagnosis, treatment, prevention, as well as surveillance in the hard-to-reach regions where malaria is prevalent. And those are primarily the regions associated with eastern Malaysia. Importantly, the government has put together some active efforts to try and reduce the potential for the reintroduction of malaria from workers that have gone to other malaria endemic regions and then returned to Malaysia. There is a risk that they could reintroduce in the regions that might have already eliminated, but there are some significant programs that have been put in place to try and avoid that. However, in the 2019 progress report, there was a cautionary note as well, in that the zoonotic parasite species, Plasmodium knowlesi, does still infect humans, and it has the potential to cause some very serious illnesses. So at present, the WHO considers Plasmodium knowlesi not to be transmitted from human to human, but this is something that it needs to be watched very carefully to make sure it doesn't get out of control. So I want to move on now and talk about some of the strategies that are required for elimination of malaria, and specifically focus on the discovery of new drugs. So the road to elimination is complex. There are many, many factors that contribute to this disease, and I've just listed the ones that I could think of and that I've gotten from the literature. Very importantly is vector control, and as I mentioned earlier, bed nets and indoor residual spraying have had an enormously positive impact on reducing the transmission of malaria. However, resistance to the currently used insecticides has now started to spread, so there's a lot of research ongoing to try and come up with new and improved insecticides. We also have seen some real improvements in the diagnostic tests that are available. In the past, we've had to rely on microscopic evaluation for diagnosis, and some of the rapid diagnostic tests that are now available, not only are they much more accurate, but they're a much quicker readout to obtain. Drugs continue to be very, very important in trying to treat malaria cases, and we also have now a vaccine. It's being studied in several different countries in Africa, and we'll see how effective that is. I think the early indications are that the effectiveness of the vaccine is really not what people had hoped for, but probably also not unexpected. And of course, commitment from government, health systems, and community, as well as funding, are all major players and major factors. So we really can't ignore any of these factors. However, moving forward in this presentation, I'm really only going to focus on drugs. I'm not going to talk about vaccines because that's not my area of expertise. So I thought it would be useful and interesting to go back and look at the historical progression of antimalarial therapeutics. So we have had, since well before the 20th century, plant extracts that have been used historically for the treatment of fever. And these plant extracts really set the stage for the isolation of a drug known as quinine in the 1940s, and that soon after led to the synthesis of the quinolines, and the most notable and most well-known of the quinoline antimalarials is chloroquine. In the 40s and 50s, chloroquine was used quite extensively to reduce the burden of malaria, and it was very effective, really dropped the death rate and the incidence rate quite considerably. However, resistance soon started to spread, and that's not uncommon with any agent that's used as a single therapy for any infectious disease. So a number of eradication campaigns that were put in place in the 50s unfortunately had to be halted in the 60s. So by the 60s and 70s, there were new antimalarial drugs that had become available, but over the next 20 to 30 years, resistance to most of these has also emerged. And then in the late 60s, there was a push from the Chinese government to isolate and identify the active component from an extract of a plant known as Artemisia annua, or the sweet wormwood plant that had been used to treat fevers in China for many, many years. And a chemist by the name of Yu Tu undertook this program. She was able to isolate and identify what later became known as artemisinin in the early 70s. And she received the 2015 Nobel Prize in Physiology or Medicine for these discoveries, and well deserved. So the artemisinins that were discovered in the early 70s are very, very effective drugs, artemisinin and a number of its derivatives, and they were used in China in the early 90s as combination treatments, and then ultimately the first artemisinin combination therapy known as Coartem was launched by Novartis in 1999. And this is a combination that contains an artemisinin derivative known as artemether, as well as another Chinese drug known as lumefantrine, and these drugs are still used today. Also in 1999, there was a formation of what's known as a public-private partnership known as the Medicines for Malaria venture. And MMV was formed to really try and take drug discovery and development activities into the 21st century, and really to address the lack of commercial incentive that was present for the pharmaceutical industry to be engaged in R&D efforts in this particular area. And I'll come back and talk more about MMV a little bit later. So into the 2000s, in 2006, the World Health Organization recommended artemisinin combination therapies, or ACTs, as the first-line therapy for all endemic countries in which malaria was present. And this really had a very profound impact on reducing the mortality and the incident rate. And then over the period of 2009 to 2018, there have been about 10 new medicines that have been launched. Several of those have been new fixed-dose combinations of different ACT combinations, and we need different combinations to try and avoid the spread of resistance. We've also seen pediatric ACT formulations that have been launched, and that's very important because kids are a major patient population. Artemisinin was launched for the treatment of severe malaria. Prior to artemisinin, quinine was the drug of choice, but it's associated with some side effects. We've seen sulfadoxine, pyrimethamine, and amidiaquine as a fixed-dose combination that was launched for seasonal chemo prevention, which has been very effective in parts of Africa. And just last year, in 2018, tafenoquine received approval from the US FDA for the treatment of radical cure of P. vivax infections. Over the period of 2012 to 2019, there have been 5 new medicines that have advanced into phase 2 clinical development. And this largely reflects some of the drug discovery activities that MMV supported in the early 2000s. So moving forward and thinking about drugs for the future and drugs that we have today, we need different drugs to address these different challenges that we face in malaria. And specifically, we need drugs to take the malaria burden from high burden to low burden, and we need different drugs to take the burden from low to zero and then to maintain zero. And over the past 10 to 15 years, the focus has really been on trying to manage cases of clinical malaria, make sure that we had good drugs for treatment, as well as prevent malaria infections in the very vulnerable population, specifically children and pregnant women. So in this portion of the control campaign, the focus has really been on optimizing the use of the medicines that we have available, making sure that the quality was there, that they all met stringent regulatory requirements, and that there were a number of different combination therapies available for use. Going forward, the new medicines that we need have a somewhat different focus. So going forward and thinking about eradication, we really need to be thinking about clearing all forms of parasite from the human host, and in doing so, interfering with the transmission cycle that inevitably occurs. And then eventually we need to get to the point that we can protect malaria-free populations from then reintroducing the disease back into those groups. So we need new medicines that treat multiple stages, and the focus going forward is really on improved patient compliance, ideally with a single dose administration. So why are these challenges so complex? And the reason is that the malaria parasite undergoes a very complex life cycle. And you can see on the diagram on the right, this is quite complicated. There are 2 stages, the human host and the mosquito vector. Within the human host, there are a number of stages, and really only one of those stages, the blood stages, is associated with symptomatic clinical malaria. So this is the period of time when people develop fevers and they report to clinics for treatment. But there are other stages of the malaria parasite that remain asymptomatic yet contribute to that progression cycle. So in order to effectively intervene with this disease, we have to be able to treat all the different stages of the life cycle, and drugs act differently on different stages, so it really complicates the issue. Potentially, drugs also act differently on different parasite stages, so what works on different parasite strains, what works on one strain may not actually work on another strain. So I mentioned before the artemisinin combination therapies, and these are definitely the most effective drugs that we have today. They are comprised of an artemisinin derivative, which has a relatively short half-life within the body, and a longer-acting compound, currently either a 4-aminoquinoline or an amino alcohol. And together, these drugs are very, very fast-acting. They're very efficacious. They act against multiple parasite stages, and there have been reported to be greater than about 98% effective in pivotal clinical trials. Importantly, they're also very stable medications, and that's important. As we know, malaria is a disease of the tropics, so there's no point in having a medicine that doesn't have a very long shelf life. And they're also very affordable, which is important because many of the regions that have malaria active transmission are also regions of poverty. However, the ACTs are now threatened by drug resistance, and this has unfortunately arisen and spread throughout regions of Southeast Asia. It was first reported in Western Cambodia in 2008, and now delayed parasite clearance has been reported throughout regions of the greater Mekong sub-region. And this drug resistance is due to not only the artemisinin component, but also the partner drugs. So we have really quite a severe situation. Many contributing factors that have led to this situation. So we can't afford to be complacent. We desperately need new drugs. We need new drugs that accomplish the treatment requirements that we have to meet, to treat drug-resistant malaria and also to improve compliance. We need new medicines that can be used in vulnerable populations, and we need medicines that can contribute to the elimination and eradication agenda. So to address some of these challenges, global partnerships, which combined the expertise and the skill sets that exist within academia, the pharmaceutical industry, as well as endemic countries, have really shown the greatest promise for success. And one of the best known partnerships is a product development partnership known as the Medicines for Malaria venture. As I mentioned, MMV was established in 1999. They are a Swiss foundation. They're based in Geneva, and US charity. And as a product development partnership, or PDP, MMV brings together academic groups, government and non-government organizations, philanthropic groups, as well as private industry, with the goal of really discovering, developing, and delivering affordable, effective, new antimalarial therapies. And you can see on this slide the integrated global partner network that MMV has established, and approximately 50% of those partners reside in academia and another 20% within the pharmaceutical industry, and the remainder spread across the other different sectors. So one of the things that MMV has been very instrumental in doing over this time period is to establish centers of excellence and centers of expertise around the world. And this is required because the parasite is very complex. We have multiple life cycle stages, all of which we need to be able to study and test the effectiveness of drugs against. And all of these skills don't reside in any single institution. So they have set up testing centers and centers of excellence around the world to make sure that we have the tools available to be able to address these challenges. They have also elicited expertise and input from a number of sites around the world that can contribute to industry standard drug discovery efforts. So for those of you that are not familiar with drug discovery, this is a chevron that is often used to try and describe the process of taking a compound from early target identification, so very, very early biology, through screening activities to identify an active compound and confirm a hit, to take that hit compound and convert it into something that's a bit more drug-like, which we call a lead, and then to take that lead and optimize it further such that it has all of the characteristics and requirements that we need for it to progress into the regulatory-driven safety studies that are needed to support clinical development. This is a very complex pathway that can go for as many as 15 years and cost quite a lot of money in order to take a compound from screening into the clinic and ultimately into registration. It also requires a vast range of different skill sets, and outside of the pharmaceutical industry, it's almost impossible to find all of these different skills in one single organization. So the box that's highlighted in the bright blue, well, it's bright blue on the screen, known as drug metabolism and pharmacokinetics, or DMPK for short, is the area that my group works in. So, looking at where drug metabolism and pharmacokinetics fits into this cycle, we actually start contributing to programs around about the hit to lead stage. At that stage, we start to work with chemists to assess the liabilities of drug candidates, to understand what structural modifications might need to be made. But the most of our activity actually occurs in this process that we call lead optimization. And what that actually is a cyclic process where medicinal chemists will work with parasitologists, DMPK scientists, toxicologists, and malaria biologists to try and optimize the structure of those compounds so that they have the required properties that they need for further development. This is an iterative process. It can go through many cycles. It can take as many as 2 years, maybe even longer, to ultimately focus in and narrow in on the best compound to progress forward. Because once we move into preclinical development, the cost of the studies that need to be run goes up quite substantially, and certainly we don't want to progress compounds that are not that are substandard into clinical development, and that certainly would not be supported. So with that, I want to move on and talk about a couple of specific drug discovery projects that my group's been involved in over the past 20 years. And the first of those is a project that's known as the Synthetic Peroxide Discovery Program. And this is a collaborative, an international collaboration with the University of Nebraska Medical Center, the Swiss Tropical and Public Health Institute, our group at Monash University, the Monash Institute of Pharmaceutical Sciences, all supported with development guidance and expertise through the Medicines for Malaria venture. And this is just a picture of the core project team. I think was taken back in about 2006, if I remember correctly. So I mentioned again earlier the Artemisinin derivatives. These are quite amazing drugs, and from a chemistry perspective and a structural perspective, they're very much unique. They contain this peroxide pharmacophore, which you don't find in probably any other drugs, certainly not any drugs that I know of. And that peroxide bond is required for biological activity. So if you don't have the peroxide, these drugs aren't effective and they don't work. And the peroxide is what gives them this unique activity against multiple stages of the parasite life cycle. But they do have a short half-life within the body. They have to be used in combination, not only for trying to reduce the development of resistance, but also such that they're effective within a reasonable treatment regimen time frame. The current ACTs, most of them require twice daily dosing for about 3 days, and that's not very long, but even that sort of treatment regimen results in compliance issues. And the starting material has to be isolated from a plant source. So you can synthesize the artemisinins. However, the synthesis is very long and it's very expensive, so the starting material is still extracted from the plant source. And back when this project began, there were considerable concerns about the availability and reliability of the plant source because it's very subject to seasonal variations in crops. So having a fully synthetic peroxide was considered to be a real advantage. Some of those issues have since been addressed. And as I mentioned, the resistance to these compounds is spreading. So back in the late 90s, this guy that's shown on the right by the name of Professor Jonathan Venestrom, who's based at the University of Nebraska, he's an excellent medicinal chemist and a very creative one that took knowledge of the peroxide pharmacophore that's present in the Artemisinin derivatives and combined that with a novel co-ozonolysis reaction that had been published in the late 90s and synthesized a series of molecules known as the 1,2,4-trioxylanes or ozonides. And you can see this structure of one of the early compounds known as OZO3. Jonathan was working with a guy called Hugues Mathiel, who still is, a biochemist at Roche. He was on sabbatical at the Swiss Tropical and Public Health Institute, and together the two of them were able to demonstrate that this series of molecules was very potent and very fast-acting, with activity comparable to the Artemisinins, yet being fully synthetic, and had activity against multiple parasite stages, so some very promising chemistry and biology. However, they were very, very insoluble molecules, and they were not active at all after oral dosing. And when you're trying to improve compliance, you want to ideally be able to give an oral therapy. And it was likely that this was due to poor pharmacokinetic properties. So through a series of associations and connections, a project team was put together consisting of medicinal chemistry, parasitologists, malaria biologists, and DMPK scientists, which was our group. We applied to MMV in the first call for proposals that they had back in 1998. The project was successful, and it actually began work in early 2000. And the starting point, as I mentioned earlier, this compound known as OZO3 was actually a very, very potent compound as a starting point. Most chemistry programs don't even come close to starting with a compound that is that active. So 1 nanogram per mil, low nanomolar potency in an in vitro assay. But it had very poor physicochemical properties, poor pharmacokinetics, and no in vivo efficacy because of those characteristics when it was given after oral administration. So through a process of lead optimization and hit-to-lead progression, we were able to come up with this molecule known as OZ78. And this was really a breakthrough molecule because it was the first compound in the series that we had looked at that had improved physicochemical characteristics. It had pretty good pharmacokinetic properties, so it had a good exposure profile when given in disease models. However, the potency had really dropped off, and the in vivo efficacy was really not what we wanted or expected it to be. We later came to know throughout the work on this series that the compounds that contained a weak acid functionality were not nearly as potent as compounds that contained a weakly basic functionality. So lead optimization on this program continued. This compound known as OZ209 was discovered. This was a very potent compound. In fact, one of the most potent compounds that we ever looked at. Had very good pharmacokinetics, but it didn't have a great toxicity profile, so that was really not acceptable. And eventually we came up with this compound known as OZ277 that ticked most of the boxes that we wanted it to. It was very potent, had good phys-chem properties, good pharmacokinetics, a good in vivo exposure and efficacy profile, and a pretty good tox profile as well. So this compound was progressed. The results of this early discovery program were published in Nature. So taking a compound such as dihydroartemisinin, so the semi-synthetic artemisinins, which required a 3-day treatment regimen, and introducing new chemistry and lead optimization, ultimately got us to the point of having a completely synthetic ozonide that had similar activity to the artemisinins and a marginally improved half-life. And that compound later became known as arterilane. This compound was licensed and taken over and developed by an Indian pharmaceutical company, and it was marketed in 2012 as a fixed-dose combination with piperequine. Phase 2 clinical trials suggested that this compound had comparable efficacy to artemether lumefantrine or Coartem, which is one of the most widely used ACTs. So during this development phase, the discovery team was no longer involved in this process, but we had continued efforts looking at the chemical series that we had, really trying to focus in on single-dose administration because this would have a profound impact on improving patient compliance. Going through a similar process of optimization, looking at structural modifications and really trying to understand the mechanisms involved in the short half-life, we ultimately discovered this compound known as OZ439, which later became known as artemisinin. This compound was taken over by MMV, was progressed into clinical development. In a phase 1 study, it showed a very long half-life with a half-life in the phase 1 study of about 40 to 60 hours, and in subsequent studies has shown a half-life even longer than that. It was quite well tolerated up to a dose of at least 1,200 milligrams, and when it progressed into phase 2 clinical trials, it showed rapid clearance of P. vivax and P. falciparum infections. And it's being developed as a potential for single-dose cure in combination with a longer-acting agent. This work was published in PNAS in 2011. So what does this mean when you think about a longer half-life? I think a graph illustrates that concept a little bit better. And this is just a simulation of what you might see with dihydroartemisinin given as 120 milligrams twice daily. And you can see the twice daily dosing over a 3-day dosing period. And by the time you get to the 3rd day, concentrations have fallen down below the target concentration range. So this is one of the reasons that we have to give these drugs with a longer acting partner compound. So with OZ277, you can see a marginal improvement in the half-life, sufficient to support once daily dosing, but still would be required to be given in combination. And ultimately, OZ439 as a single dose administration with a considerably improved half-life, having much, much greater exposure. So the potential for single dose administration is really based upon this improvement in half-life. It would still be a combination therapy, in order to decrease the possibility for resistance development. So currently, this compound is in phase 2B clinical trials through MMV, and it's been combined with a compound known as Ferroquin, which is a Sanofi compound. In 2015, this program was highlighted by a report that came out from PATH that brought attention to 30 high-impact innovations that had the potential to save lives. So this was, we knew nothing about this, and it was a really good thing to actually see. Okay, with that, I want to move on to the next drug discovery project. And this is a program that was targeting pyrimidine biosynthesis for the treatment and prevention of malaria. And this was a collaboration, it still is a collaboration, being conducted through the University of Texas Southwestern Medical Center, the University of Washington, our group at Monash, MMV, GSK were involved at some of the early stages. They contributed to the chemistry program. The pharmaceutical company Takeda is involved in the development program, and this project has been funded through the NIH in the US since 2007. And this is a picture of the core project team that I think was taken in around about 2010. So this target that we were trying to inhibit is known as dihydroorotate dehydrogenase, and that's a bit of a mouthful, so I'll call it DHODH for short. This was a novel antimalarial target, had not previously been attempted as a target for the treatment of malaria prior to this project. The reason this was a very interesting target is that DHODH is an enzyme that's present in the pyrimidine biosynthesis pathway, and in parasites, de novo synthesis of pyrimidines is required for growth and development and to maintain survival. And the reason for that, of course, is that pyrimidines are the building blocks for RNA and DNA, and without RNA and DNA, the parasite cannot survive. So if we inhibit this pathway through inhibition of the enzyme, then ultimately the parasite should die, or at least that's the hypothesis. The other important thing about this program is that DHODH is an enzyme that's also present in human cells as well. And if you inhibit human DHODH, you can end up with some certain side effects. So it's important that we had selectivity for the parasite enzyme over the human enzyme. So the structures and amino acid sequence in the active site of this enzyme suggested that it should be possible to get selective inhibition. But this was a very important part of our overall testing strategy. So this program was initiated by Meg Phillips, who's a professor at the University of Texas Southwestern Medical Center. Meg is a biochemist and structural biologist, and she and her team conducted a high-throughput screen against the purified Parasite enzyme and also included a counter screen for the human enzyme to identify some hits against this target, and then went on later to confirm that those hits were due to inhibition of DHODH, the target that we were trying to inhibit. And this is the first compound that emerged from that screen. It's known as DSM-1. DSM stands for Dallas, Seattle, Melbourne. We weren't terribly creative in those days, but that's what we named it. And you can see from this graph in the middle that there was no activity against the human enzyme, which is what we wanted, but quite potent activity against the parasite enzyme. And for an initial hit to have an IC50 in the range of 50 nanomolar is actually very encouraging. It also had activity against the parasite enzyme at a concentration of about 60 nanomolar, so very consistent with the enzyme. This diagram over here on the right shows DSM-1 bound in the active site of the DHODH enzyme. So this was a very good starting point. It had good potency and good selectivity, but had poor metabolic stability, a poor plasma exposure profile, and no in vivo activity when it was administered orally. So we embarked on a lead optimization program to try and reduce metabolism, and we also wanted to try and improve potency. We didn't do too well in the first round of iterations, and we came up with this molecule known as DSM-74 that was actually about 5-fold less potent, but it had an improved pharmacokinetic profile. It was much more metabolically stable, had a much more sustained plasma exposure profile, and it allowed us to take a compound into an in vivo model of disease to see if inhibition of this enzyme actually works. So it provided a proof of concept test as to whether or not inhibition of DHODH was actually very useful. But because this compound lacked potency, it was really only a partial response that we saw in that molecule. So the optimization process continued, really with a focus on improving potency without sacrificing the pharmacokinetics, and eventually this compound known as DSM-265 was discovered, and this was the final candidate. It had very good potency down in the low nanomolar range, good selectivity, so it did not inhibit the human enzyme, sustained plasma exposure, and very potent in vivo activity. And the results of this program were published in Science Translational Medicine in 2015. So the data that was generated here supported the progression of this compound into preclinical safety studies, scale up and formulation. It ended up going into a phase 1 clinical trial through the Medicines for Malaria venture, where it was shown to have a very well-tolerated profile up to quite a high dose. And importantly, it had a very long half-life, a half-life of around about 100 hours, which is very important when you're trying to target a single-dose administration. The compound also progressed into phase 2 efficacy trials in Plasmodium falciparum and Plasmodium vivax patients, and it worked really very well against Plasmodium falciparum, and in these patients it showed a greater than 90% effectiveness, which was very encouraging. However, it did clear parasites, but it was not curative against P. vivax infection, so that was disappointing, and it's still something that we don't completely understand today, and it's something that we would very much like to understand but don't. So this compound is still in clinical development, and currently there are discussions ongoing within MMV around a suitable partner drug and whether or not this molecule is better treated for treatment or for chemoprevention. So this is just another example of another program that we've been involved with that's led to successful candidates going into clinical development, all supported through this product development partnership model supported by MMV. So On this slide, this is a diagram of the global malaria portfolio. I've downloaded this from the MMV website. If you're interested, you can go there, and there's a lot of information on the website, which is very interesting to look through. And this just shows you where those 3 programs that I've spoken about sit within that product development pipeline. There are a number of other compounds, obviously, in clinical development, and what's not shown on this is all of the early discovery stage programs that are also being explored. So many activities all trying to find new antimalarial drugs. So with that, I want to just finish up with a couple of very simple slides, but with, I think, some very powerful messages. I think many of you would be familiar with the ancient African proverb that says, "It takes a village to raise a child." So what does it take to eliminate malaria? Well, I think first it takes a vision for a world that doesn't have any malaria. And I particularly like this quote from Melinda Gates back in 2007. "To reach a day when no human being has malaria and no mosquito on Earth is carrying it." That's the kind of vision that we really need. We also need a goal, because a goal and targets really set the agenda for elimination and ultimately eradication. We need a plan to be able to deliver the objectives of that goal. And we need multiple strategies that all fulfill the goals of the plan. And sitting behind this, we need people, and we have people, committed people around the world working in different areas with different skill sets, all trying to discover and develop new drugs, working as a global village to try and address some of the many challenges that we have to eliminate malaria. So with that, I will close and say thank you very much for your attention. Thank you very much.